Hello, everyone. Thank you for joining today's DoD Environmental Planning and Conservation webinar series presentation. As a reminder, today's presentation will be recorded and then posted on the Natural Resources DenX website under Resources. Today's presenter is Giri Athrey, an assistant professor in the Department of Poultry Science at Texas A&M University. He will be discussing assessment of the population genetics of golden cheek warblers, updates from genome-enabled studies. Following the presentation, we will have time for questions. Before we get started, please take a moment to make sure your phones and web browsers are on mute. And with that, I will turn it over to Giri.
Thank you so much. Really happy to give you this update on gold cheek warblers. So first of all, I'd like to introduce myself. So my name is Giri Athrey from Texas A&M University. And my two main collaborators on this work were Dr. Laurel Moulton, from a biologist in the city of Austin, and also Lisa O'Donnell, who's also a biologist in the city of Austin. And many thanks to our funding for this work, which was the DoD Legacy Program was instrumental in this, and we were funded through a cooperative agreement and received funds through the U.S. Army Corps. So many thanks to that. So a brief introduction on the golden cheek warbler. I think many of you are familiar with the golden cheek warblers and generally the story of these endangered species. Golden cheek warblers or Setophaga chrysoparia are endangered birds which breed in central Texas, and you can see on the map on the right hand side most of their breeding range is in this crescent-like shape, which spreads from southwestern Texas and into the northern region to some extent, whereas they winter in central parts of Central America. And what we do know is that one of the it's one of those sentinel species that have been a poster child for conservation for a long time. And at the time of their listing, we know that like many endangered species that were listed, they were pretty scarce in number, and a few hundred breeding pairs were supposed to have existed. But one of the things with many of these types of scenarios is that we don't really understand how abundant they were in the past. So it is hard to sometimes determine whether these species were historically abundant, where they kind of specialized and having a small habitat, footprint and a small population size in their history, or whether they were extremely abundant. So these are things that we cannot always have good answers to. And many of the population genetic studies are essentially in a nutshell, trying to understand these types of questions. And if they were abundant, how abundant were they? And if they actually declined in size over time, what are the impacts on the genetic diversity and viability of these species? And how do we go from here knowing that information? Similar to other conservation genetic studies, this one was motivated by these same kinds of questions as well. And the other context to this is obviously the management efforts, that have been focused on the golden cheek warbler. So you can see here, for example, there is some documentation on the habitat that's managed. So on the right-hand bottom side of this graphic, you can see how much of the area it's expected to be in 2016 versus in 1990, and that can be down to both efforts to protect the species habitat and also discovery of habitats that were probably previously unknown. And similarly, some of the modeling studies have, based on the habitat suitability, modeling studies have projected that at least by 2016, that the populations had risen to a much higher number than during the time of listing. So it went from, say, 13,000 approximately to 260,000. So a very large increase in the population size. Now, we also know that during this time, for example, this region of central Texas has seen an enormous increase in land conversion and habitat change. If any of you live in this area in Texas, you'll know exactly what I'm talking about. And just between 2001 and 2011, there was an estimated 30% reduction in warbler breeding habitat based on, again, studies by Texas State University. So one of the things which are very difficult to reconcile with these types of information is how do we accurately measure population recovery, the numbers of the species, and ultimately also additional considerations such as what kind of protections need to be afforded to the species based on other metrics such as their genetic viability. And as we know in many ongoing conservation projects, the consideration of genetic diversity is one of the important criteria that needs to be included. So previous genetic studies showed a decline in genetic studies. So this is again some historical background on what we already know about the species. And the previous study was also conducted partly by myself and my colleagues, more than a decade ago. And these asked at that point, how did habitat changes and population size changes impact genetic diversity? And our rationale at that point was we need more historical context to answer these types of questions. So in a study we published in 2012, for example, we used a mixture or combination of ancestral or historical museum-based specimens to contemporary populations. So these were collected over 100 years apart to try to answer questions such as how much has genetic diversity declined over this 100-year period? So the time spanning what we know to be the lowest point in, at least the recorded lowest point in golden cheek warbler history. So I have these two plots here, which are kind of talking about the expected heterozygosity, which is a measure of genetic diversity. And in these studies, we compared three populations or kind of areas which were at the county level. So Kerr County, Bexar, and Travis are these counties in Texas, which fall in some of the prime habitat for the species. And over this 100+ year span, we see that, for example, heterozygosity is what declined quite a bit. And similarly, allelic richness, which is a slightly different kind of a measure of genetic diversity, also showed statistically significant declines. Now, there's some difference in these two measures. For example, so heterozygosity declines much slower compared to allelic richness, which can respond much faster to population size changes. So they speak to different kinds of forces acting on these populations. So I'd like you to keep this in mind because we'll be revisiting the topic of heterozygosity later on. But anyway, these were among the first studies on this topic, and which showed that over the preceding century, golden cheek warblers had experienced quite a decline in their genetic diversity. Now, of course, this is the data was collected between 2002 and 2007, and we know that in the last 5 to 6 years, there have been new estimates for the population sizes of the species. So one of the things we want to know is, now that the population seem to be on an increasing trajectory, do we also see a recovery in the genetics? So we'll get to that. Now, one of the things to obviously make clear is why we care about genetic diversity is that it is quite important in terms of a species' ability to survive and even thrive in their existing environment, let alone any kind of future pressures or stressors they may encounter. So the greater the amount of genetic diversity, they have a greater resiliency to recover from setbacks. So it could be events such as the very bizarre ice storm that Texas experienced in 2021, for example, and birds are already arrived, and we know that many migratory species had a very adverse mortality event thanks to those kinds of things. So the ability to survive, one-off or repeated stressors can be down to can be quite influenced by the genetic diversity. So that's why we care about measuring and ensuring that there is sufficient genetic diversity, or if there's not, how we can manage and improve those metrics. So here's just an illustration of what happens to genetic diversity. You go from this, this container which has many colors, which represent the diversity of the genetics or the allelic diversity in those populations, and through this random selection process, you end up with new populations which have much limited genetic diversity. Now, the same study also looked at patterns of gene flow between species. So in this case, we used a measure called the FST, which is essentially looking at pairwise measures of gene flow. And this measure is, as you can see on the y-axis here, it represents the FST values. So low values of FST means that there's actually high gene flow between pairs of populations, whereas high values or higher values of FST indicate that there is actually reduction in the amount of gene flow or mixing between populations. So you can see that for each of these maps here, I have a pair circle. So it would be Travis and Kerr County, or Travis and Bexar County, and so on and so forth. So those three pairwise comparisons. And we can see that in the historical period, there appear to be much more connectivity and gene flow between these pairs of populations, whereas these had already declined quite significantly by the early 2000s. So this is a brief background of what we already knew about this species. But like I said, this is dated information, and with the upcoming review of the species status, we proposed, and we were also tasked with answering these questions and reviewing this information based on new and more comprehensive genetic technologies. So, in the first objective of our project was to generate a new genetic resource which is not present for the species, and it is to create a de novo, or from scratch, a genome reference. And we use a combination of new sequencing technologies to enable this, and this will become a freely available public resource which can be used for future studies, and it standardizes all future genetic studies of the species. So we don't have to worry about, oh, can we compare data from 20 years ago to the contemporary technologies from here onwards, of course, because they will all be based on the reference genome. So what is a reference genome? So a quick crash course on this. Reference genome is a haploid representation of a species genome. Many of you may have heard of the Human Genome Project, of course, and that's where the advances from that has now spread far and wide to many model and non-model organisms, including many species of conservation interest. And I was told that when we complete the reference genome, this will be, if not the first, one of the first reference genome for a U.S. endangered species, which had been funded through federal sources. So, so this is quite a leap for the species on its own. And typically, the genome reference is constructed from a single individual, which is what we did in this case, an individual caught from Travis County, and alternate alleles, because vertebrates, including birds, are diploid organisms, they can have two alleles. So one of the things that we want to know is our ability to recover these genotypes, so we can have better representation or understanding of the kinds of genetic diversity or heterozygote loci that we might come across when we survey an entire species. And now there's various stages in which genome references can be polished. So obviously the best is to have them organized into chromosomes, which is very difficult and not something people achieve in the first go. So again, for reference, I mentioned the Human Genome Project, which was first sequenced in 2001, but the first telomere to telomere human reference genome came available in 2022. So it kind of tells you that how much of a process this can be. But that doesn't mean we have to wait that long to be useful. I'm just talking about the level of polishing that this might take. And the next best level is to have genomes which are at the scaffold level. And 95 to 98% of all genome references available out there are going to be in this category. And then the third and probably the most rough draft of a genome is in the contig or supercontig level. And so to kind of give you an understanding of how genome assembly works, we take copies of genomic DNA by taking a blood sample or a tissue when possible. The DNA is homogenized and turned into this pile of shredded bits of DNA. And from then we have the option, or we have the challenge of putting together a nice readable copy of a genome, or you can think a better analogy, maybe a book where there are chromosomes which are organized as chapters in the book. And so this is the analogy that we can use for how the genome works, or the genome assembly works. And so in this case, we'd be using something called whole genome sequencing to generate a reference genome. So if this were the actual genome sequence, the top line, which is represented in blue, we generate sequencing data, which is in these bits of fragmented pieces, which are sequenced reads. And ideally, we end up with a perfect assembly, but like as I just told you, that is a very, very difficult and long process. Whereas the typical assembly ends up being in this dark blue, which is a scaffold level assembly, where you're going to have some gaps in unknown sequences, mostly comprised of repetitive regions of the genome, which may not always be most informative. So it is not that we are going to be missing a huge amount, but it is just a more challenging part of the genome to assemble. So what exactly did we do in this case? So we, as I mentioned, sampled a single individual captured in Travis County, and we used two technologies, a short read technology called Illumina sequencing, with 150 base paired sequences, which reads every fragment of DNA in two directions. And we also combined it with long reads, which can range from anywhere from 5,000 to 100,000 base pairs. In this case, called long read sequencing with Oxford Nanopore technology. And here you can see what exactly happened. So as I told you, the first one is our contig assembly, which is called the Hassler hybrid assembly. You can see the number of contigs, or these are individual pieces which can be assembled with very good confidence, has a lot of pieces. And the largest contig in this case was 4.5 million base pairs, which is good contiguity. But if you think about this, a typical bird genome can be anywhere from 900 to 900 million to 1.2 billion base pairs. So the contiguity scores can be better, and in many cases should be better. And the N50 is a metric we use to kind of also describe how good are the pieces that we've assembled. So what we want is the N50 values, for example, to be as big as possible. So in this case, we can see here we went from the Hassler hybrid assembly to a scaffolded assembly to a gapped scaffold assembly, which is the best we could get to with this data. And it has 2,450 pieces, although these birds probably have something like 38 to 40 chromosomes. These are not assembled into chromosomes because this is a scaffold level assembly. And the longest contig you can see improved by 3 times through our bioinformatic processing. And the N50 is 1.5 million base pairs. That means over 50% of all the reads or the scaffolds in our assembly are at least 1 million base pairs in length. So that means we have very high contiguity of our assembled genome. And we ended up with about 817 million base pair reference genome. And like I said, this is a really good draft assembly. It compares very favorably to most draft assemblies because of the two or the hybrid technologies we've used in this case. And it has very good contiguity scores. Now we can measure this in other approaches also. One of them is called the BUSCO approach. And this stands for Benchmarking Universal Single Copy Orthologs. That means we want to know if the assembly was able to reconstruct the core orthologous sequence that are known from all bird species. And in this case, we found that there's actually a very high proportion of complete single copy orthologs, about 70%, which were recovered, whereas some of those orthologs are missing in this case. And that is sometimes this is again one of those things that are improved with subsequent versions, including additional population data. So this again doesn't mean that it is a bad assembly or anything. It is just that it shows that the first draft or first assembly, this is a pretty good assembly, and we recovered 75% of coding sequences that are found among all birds in this assembly. So now that we've established that we have a very decent, very good reference assembly, we're able to use this as the base for building population genetics and surveys in this case and in future studies, for example. So the second objective of this work was to perform a genetic assessment of the golden cheek warbler populations, their diversity and their structure, and population size history across the breeding range, and use this information or provide this information for the upcoming status review of the species. And so, as I mentioned, the rationale for this was to update the work done previously by a colleague of mine, Denise Lindsay, who is now with the U.S. Army Corps in Mississippi, and my own study, which I also referred to earlier, which did measure genetic diversity, but we wanted to obviously update this information with whole genome data. So what did we do in this case? We performed probably, well, at least for the genetic study, the most extensive sampling for the species. We sampled across 13 counties, which are depicted on this map here, which represent actually 9 sites because for example, Belle Coriel, which is Fort Hood, is considered as a single site in this case, and similarly Kinney Edwards, which are kind of also adjacent, and the actual population sits across these 2 county boundaries. And most of the work was conducted in the summer of 2019 and 2020, and a little bit in 2021. And overall, we collected across these, you can see here the table of the samples and the sample numbers that we collected. So across these 11 counties, or as I mentioned, so 13 counties, but just to draw your attention again, Kinney Edwards and Belle Coriel, for example, are listed together. You can see the numbers of individuals we sampled from each of these, yielding a total of 282 samples. And we did sequence, or we attempted to sequence DNA from all these samples. However, 43 of these samples had very poor template quality. because we are, as you can understand, we are collecting 1 to 2 microliters or 1 to 5 microliters of blood in many cases from a single brachial prick, and it's not always sufficient quality or quantity to proceed with whole genome sequencing. So 43 of these samples could not be sequenced in this particular study. And with the remaining 239, we generated short paired end sequencing data, again, sequencing on the Illumina platform. And for each of them, we generated 12 to 15x genome coverage. Now, what this means is that based on how many reads of sequencing we generated, we have an average coverage, or we have observed every single base pair in the genome between 12 and 15 times. So that means we have a very good understanding of whether a particular nucleotide polymorphism, or a SNP, for example, is actually a real variation or an error. In this case, this amount of sequencing coverage at very high quality data basically obviates or completely removes the chance that we have erroneous sequence data or poor quality data, which may be affecting any of our calculations later on. And this is also, as a comparison, a few years ago, I participated in a golden cheek warbler, a black cap vireo sequencing study, where we only achieved 2x coverage. And so many molecular ecology and wildlife genetic studies use much lower coverage for various reasons, because of budget reasons in some reasons, or mostly budget reasons. So what we have here is a very high amount of data, which allows us to answer more in-depth questions, including the kinds of haplotypes that we might encounter in these populations, and investigate deeper questions to do with whether we have specific differences among genes of functional importance, such as immune gene diversity and so on and so forth. That's not part of what I'm presenting today, but again, this data was collected and generated with the idea of investigating more of those deeper questions. So in this case, though, we analyzed the genetic diversity and structure with open source bioinformatics tools, and all the analyses that are performed on the supercomputers at Texas A&M University. And so let's get into the results from this. So what we have here, first of all, is a genome-wide estimate of nucleotide diversity. So this is a very commonly used measure of diversity, also called, notated as pi. And you can see here that for golden cheek warblers, we have a genome-wide average nucleotide diversity of 0.0014. And how does this compare to some of these other species? This data is not new, because I had presented this, because we had this was one of the earliest data we generated, and I probably presented this to this group more than a year ago, if anybody remembers. So this kind of compares to many other species, in some cases favorably, some cases not so much. So for example, you can see that on the top right, you have the Physadulla flycatchers, which are a quite abundant species, and we have golden cheek warblers, which have about a third of their nucleotide diversity values. Also the red jungle fowls from Southeast Asia, we have about a fourth of their genetic or nucleotide diversity values. Similarly for these sparrows, but on the same level as hooded crows. So nucleotide diversity can indicate, the total or the amount of genetic diversity generally at the genome level. And it has been used by in many cases as a way to classify whether a species is at risk or not. And in this case, they are not extremely low, but they are on the lower side compared to many other abundant species. Now, the other genome-wide metrics we looked at was, first of all, the average heterozygosity. So in this table, we can see a site-by-site estimate of the average heterozygosity. So in this table, you see on the second column, we have the sample size. So we have very good sampling in every single case, over 10 individuals or over 15 individuals in every case. And the third row has the average inbreeding coefficient, which I'll talk about in a second. And the last column has the average heterozygosity or observed heterozygosity. So what you can see here is the heterozygosity values are also small in this case. Now, this is much smaller. These numbers are going to be much smaller than what you may have observed using other second generation genetic tools such as microsatellites, because here we're looking at whole genome averages. So observed heterozygosity values from whole genome data tend to be much smaller than marker data, but these are still very small, showing that we have low heterozygosity in these populations. Now, what's actually worrying is the inbreeding coefficient, which is the FIS. So FIS values can range from -1 to +1, and the positive values tend to indicate more inbreeding. So for example, an inbreeding coefficient of 1 indicates that you have a fully inbred population. So any values that are moving farther away from -1, and especially positive values, are not good news for inbreeding. So a fully outbred population would have values nearing -1. So we can see here that all these populations show positive and over 10% of inbreeding in many cases. So for example, probably the worst one on this list is Travis County, which you can see has a 12% inbreeding coefficient. So with a species-wide average of nearly 10%. So this is actually probably not the best genetic diversity level news for the species. Now, how do we look at, or what do we look at in terms of population structure? So here, again, I'm showing values of pairwise FST. So if you recall, I showed a graph earlier comparing heterozygosity between historical and contemporary populations. Again, higher FST values indicate reduced gene flow, whereas lower FST values are going to indicate higher amount of gene flow. So an FST value of 0 means that they are a fully panmictic or well-mixed population. FST of 1 equals you have a fully separated population. So again, those are theoretical minima and maxima. We have never seen values of that nature, but obviously when we have values which are away from 0, we know that these are going to be problematic. So in this case, the way to look at this is it's a matrix of these pairs of populations, and I want you to look at these in the rows. So the coloration is not, it can, I don't want you to be misled by what the coloring means, but they are basically relative shading within that row. So for example, if I were to look at Bandera County and how much gene flow there appears to be, or differentiation there appears to be between other populations, the worst or the most differentiated in this case would be with Palo Pinto, which is kind of north of Fort Hood. So it's probably the northernmost on this map here, whereas Bandera is going to be west of Bexar County. So it is probably one of the southernmost. So so you can see across, if you scroll your eyes across this row, geographically most near populations like Uvalde or Bexar County are going to have lower FST values, whereas Palo Pinto seems to be the most differentiated. Now, so similarly, I want you to just look at what's happening with Palo Pinto. It appears that it is extremely differentiated statistically significantly from every other pair of population that we see here. And in many cases nearing differentiation or nearing or exceeding 2% differentiation. And this is again based on whole genome data. So to observe such a high degree of genetic differentiation based on a whole genome data, we must be seeing either very limited gene flow between those population pairs, or a combination of low genetic diversity and high inbreeding within those populations to drive this pattern. So obviously the how red these Palo Pinto is can tend to distract.
Giri, I think you accidentally mute yourself.
Can you hear me again?
Yes.
How long was I muted?
Oh, just for like 30 seconds.
Oh. Oh, can you hear me again?
Yes.
Okay, perfect. I don't know what happened. So basically the main takeaway here is while Palo Pinto seems to be driving the color patterns here, and obviously appears to be the most differentiated or isolated population here, we need to look at other patterns here and visualize the same thing in other ways, so we understand the population structuring in the species better. So you can see here again, Palo Pinto does kind of hang out on its own based on this ordination plot, but so does Bexar County, whereas we see there are some other populations which are much more closely related to each other. So Travis and San Saba, which are obviously one of the easternmost, and then directly northwest of Travis is where San Saba is. And then we see Bel Coriel being most similar to Bosque County, whereas Somerville is again appears to be on its own, not really well connected to anyone, whereas Kinney, Edwards, and Uvalde are not very surprisingly most close to each other. So, but to me, the most surprising part of this is how isolated Bexar County also appears to be, based on this information. And Bexar County is in relation to Uvalde and Kinney Edwards, which are the more western populations here. So now, when we do see this, one of the common questions people ask is do we see isolation by distance? Which means, do we see that populations are more differentiated as a function of increasing geographic distance? So we performed this particular analysis and a Mantel's test, and actually there was no significant isolation by distance in this case. Now there are some potentially good explanations for this in this case. We already know that heterozygosity is quite low in these populations, and as FST is driven by a large degree on the within population heterozygosities, when we have low heterozygosity values, we may not have sufficient resolution to observe isolation by distance. So this could be an outcome, but even without worrying about isolation by distance, there are some very interesting patterns in the population structuring based on this whole genome data. Now, let's come to the point of the historical and the contemporary population sizes. So the historical demography, as I pointed to very early in this talk, is something we don't usually have a good idea of, because we have no idea whether these species were abundant in the past or are quite rare. So we used an approach called the PSMC approach. It stands for pairwise sequentially Markovian coalescent. It's a coalescent-based approach which uses a single genome data and looks at the heterozygous genotypes at every given site in the genome, and tries to infer the coalescence times for those alleles, which means when exactly did two alleles at the same locus share a common ancestor, and based on that, it models the ancient effective population size of the population. So again, the effective population size, not something I defined earlier, but it refers to the size of a population which actually controls the genetic parameters in subsequent generations. So you can think of them as the number of actually breeding individuals who are going to pass on their genes. You can think of it as the ideal population which experiences the same amount of genetic drift as the census population. So there are obviously some technical definitions of what effective population sizes mean, but a simple explanation is it usually refers to the number of breeding individuals contributing to the population. So in the PSMC approach, only queries the ancient demography. So you can see on the y-axis the effective population size, and these are in 10 raised to the fourth, so in 10,000s. And on the x-axis, you have time, which is also on the log scale here. So you have ending at 10,000 years, and then 100,000, a million, and so on. So you can see here that based on this approach, golden cheek warblers were historically, so between 1 million and 100,000 years ago, not that abundant, well, relatively speaking. It had an effective population size of in the 20,000s, whereas in the last 100,000 years, they rose steadily to become much, much more abundant, reaching an effective size of about 90,000. So this is actually quite a big population size compared to many even currently abundant species. Snow geese, for example, come to mind, which have a very large effective population size. Among extinct species, passenger pigeons had very large historical effective sizes as well. So, but this particular approach stops at the 10,000 year mark. And to query what's going on more recently, we need other measures of recent effective population size estimation. And we did that using 3 different approaches. And when possible, we compare, or when we had this data, we compared them to what we had found in the 2011 study. And you can see here 3 approaches for each population. Any or effective population size estimated using the co-ancestry method, the linkage disequilibrium method, and the heterozygosity method. What we can see from here is that, again, these are what these means are they refer to an estimate of the effective number of breeding individuals within each population. So the alleles that we can survey and observe in current populations can be inferred to have been contributed by these numbers of individuals. So if you see, for example, 152, that means 152 individuals contributed to all the existing genetics or in the recent past. Now, when possible, when we compared this to the 2011 study, we see that while one of the methods shows that, say a difference of 49 versus 254, across these methods, they're all sharing their confidence intervals, essentially. So you have their 95% confidence intervals, and we see a large overlap. So we can see that they are sort of in the same ballpark, if you will, indicating that the effective population sizes are much smaller, than in the historical time. So certainly not in the thousands, and most definitely not in the tens of thousands. And not surprisingly here, we see that the heterozygosity-based effective sizes are among the lowest that we can see, and these are actually among the lowest we could find in many vertebrate species. And this is not new. This is something we had observed in the 2011 study. So what we do see here is that the whole genome data seems to support the same, similar picture, or paints a similar picture of what we found previously. Now, lastly, we also looked at genome-wide test of neutrality, which is trying to see whether the patterns of allele frequencies can tell us anything about their demographic history as well. So this is usually used as a test for selection. So negative values of Tajima-SD, so less than 0, indicate that there has been a positive selection or directional selection, whereas positive values of Tajima-SD usually means that they have either balancing selection or a recent, population bottleneck, recent severe population bottleneck. So what we see in this case is that we have overwhelmingly positive values of Tajima-SD, as you can see from the number of colored dots. So we are looking at millions of individual sites here. So these are based on windows, 10,000 base pair windows, which we scan across the span of the 870 million base pairs. And we get an estimate of Tajima-SD for every one of those windows. And each of those dots here, they represent a value from that window. And what we can see is very few of those seem to indicate Tajima's values less than 0, which again tends to indicate positive selection, for example. And the majority of them are over 0. And when we see Tajima-SD values about 2, for example, those are usually statistically significant for showing that there is a significant number of loci experiencing the effects of this demographic change, usually a severe population bottleneck. And so all of these populations show, as you can see, not identical, but similar trends of demographic history based on now the many metrics that I've shown you. The genetic diversity are low. The FST measures are showing that there's some increasing amounts of isolation in some populations and some very interesting population structuring, which are in a way unexpected. The effective population sizes also indicate that the population sizes remain small and that they haven't really recovered, or there hasn't been enough generations to recover from the population bottlenecks the species has experienced. So some takeaway messages. So we show that GCWR golden cheek warblers show a genome-wide average nucleotide diversity, which is on the lower side of 0.0014. Low levels of heterozygosity in every population we surveyed, with a species average of 0.31. There was also a high level of inbreeding within each population, which in my opinion is one of the most worrying things about what we are seeing here. And as far as isolation, I pointed out to Bayer and Palo Pinto populations showing higher degrees of isolation, and to some extent, surprisingly, less than expected amount of mixing between the Fort Hood populations and their neighboring populations. And also to some extent a lack of isolation by distance, which can be suppressed by low heterozygosity, and low effective population sizes show that these population sizes, at least genetically speaking, are small and haven't recovered from their genetic bottlenecks. Overall, we didn't find any much support for a genetic recovery, and there appears to be some increasing isolation in many of these populations. So with that, I'd like to thank our funding again, and the many people who made this work possible. I've listed them all here. I'm not going to read them all, but I really appreciate the various forms in which we received help, and I'm going to take your questions at this point.
Thank you, Giri. If anybody wants to come off of mute to ask your questions, we have about 10 minutes of time for questions and answers. Giri, is there any, one of the questions that I have is, I know sometimes that they bring in other species to help boost populations, and I know with birds that's kind of hard to do. Is there any thoughts on how that that the how you can increase the population of the golden cheek warbler within this area, within their habitat ranges, or is it going to be dependent on time?
I'd say, there's really two things to do with these types of things. Well, I think let me answer your first question. So for any sort of population rescue from other populations is kind of what you're hinting at, I believe. I think that's, unless we have some cryptic population that we don't know about, I think that is going to be challenging. I know that they have tried this in pelicans, for example, and I would say many of the known examples are going to be from among these larger bird species. I don't know if it's possible with songbirds, especially migratory songbirds. But the other part of your question, yes, I think the recovery usually takes time, and I don't have this number with me, but when we or right at the top of my head, but when we have estimates of heterozygosity, we can actually calculate how many generations it is going to require to recover if we held population sizes constant, as in not cause any further declines. And it's in the tens of low hundreds of generations. So time is one of the things that we need to protect them until they're able to get enough natural processes which recover that genetic diversity. And the other one is promote connectivity between these populations. I think that it remains one of the most important and available tools for sure. Other questions?
Hey, Dr. Athrey, this is Gary Pandolfi with Fish and Wildlife Service. Can you hear me okay?
Yes, I can.
Hey, thank you for the great presentation. Really appreciate being able to watch it today. I wanted to ask, and this is coming from a very non-genetic background, so forgive me if it's a dumb question. Given the sampling focuses primarily on male warblers and not so much the females, how does that, or does that impact the results or the potential results here?
Yeah, great question. There are so with other previous approaches, I think this kind of question has come up, but generally speaking, because we are not limiting our analysis to specific chromosomes or especially sex chromosomes, it is not something of a concern. So in birds, they have the ZW sex determination system. So the females are actually going to have the ZW kind of genotype for their sex chromosomes. The males are going to be ZZ. So we haven't really looked at the W chromosome with this data, but in other birds, the W chromosome isn't actually very functionally important. It's comprised mostly of repetitive sequences, which don't do a lot of important stuff. Now, as far as you consider the rest of the autosomes, so the regular chromosomes, we are not going to find sex bias differences in their genetic patterns. So that's basically my answer to that is. I don't expect them to be different. If we'd only limit it to the sex chromosomes, I think we would have some head scratching to do about it. But this is based on all the autosomes, so where we don't expect any differences.
Okay, perfect. Thank you for explaining that. That makes perfect sense. I appreciate the answer.
Sure. Other questions?
If anybody doesn't want to come off mute and ask, you can always type your question out in the chat. John asked, what with a geographic contiguous population like the Warblers, wouldn't you expect less diversity?
Well, I mean, if I'm understanding correctly, a geographically contiguous population. Well,
Yes.
So that's, I think that's one of the questions that we are looking at, whether there's actually geographic contiguity, and there doesn't seem to be. They seem to be not contiguous. They are showing signs of being differentiated or isolated from each other, so they are not having that much gene flow as you might expect in a contiguous population. And regarding how much genetic diversity we would expect, yes, if there was a large contiguous population, we would expect higher levels of genetic diversity, actually, not less. And we did see that trend in our previous study where from a century or just over a century ago, it appears to have been more contiguous. And there was higher genetic diversity than we see now. So the current populations aren't showing the same level of contiguity, and they have less genetic diversity because of that, because they've been isolated and they've also experienced these bottlenecks which they haven't recovered from.
Thank you. Thank you. I think we're going to end there since we have only like 2 minutes left, and I don't know if we'll be able to answer your question, the question in time. So, Dr. Giri, thank you so much for coming on and giving this presentation. It was very informative, and I wish you the best of luck with your continued research on this species. And thank you everybody for joining today's environmental planning and conservation webinar presentation. And I hope you all have a good afternoon. Bye.
Thank you.